Almi, the project staff for Bioinformatics from the Philippine Genome Center, Visayas. In this video, we're going to talk about what is bioinformatics, why is it so important, and how can we use bioinformatics to solve some of the problems that we face today. In 1799, some French army engineers who were part of the Napoleon Bonaparte's Egypt campaign discovered a stone slab containing hieroglyphics while making reference to a port near the town of Rashid or Rosetta. But why is this stone so special and of such great importance, as there were lots of ancient Egyptian hieroglyphics that were already discovered prior to that? The reason behind this is, within the hieroglyphics of the Rosetta Stone, the stone contained encryption in Greek, along with the ancient Egyptian hieroglyphics. Using this, researchers were able to crack the code of ancient Egyptian writing system, which at the time remained silent for a millennia. Similarly, every one of us has a book of fate of our own. Everything about us is already written in the book, whether that may be our gender, the height of which we can grow, what skin color we have, which disease we are immune to, and which disease we are susceptible to. This book is our genome, or the DNA. We discovered DNA more than 150 years ago. We know that it is made up of nucleotide-based sequences, and it is huge. The human genome contains approximately 3 billion base pairs. DNA contains the blueprints for every biological process of every living thing on Earth, and even a small defect in the genetic code can cause devastating health consequences. Understanding how genes work, both individually and with each other, is crucial to the development of 21st century personalized medicine. However, the problem is that even when we actually have an organism's DNA in front of us, it is often not easy to tell what an individual gene does. Even more difficult is to tease out the subtle influences that multiple genes have on each other. But answering those kinds of questions is foundational to attacking genetically influenced disease at the root cause. Today, new high-throughput technologies bring unprecedented opportunities for life science research. They allow us to get new data never seen before, to study new questions impossible to study before, and to discover new phenomena unimaginable before. Now, we can sequence not only one person's genome, but also many different people's genomes. These allow us to study the genetic differences between different people at genome scale, as well as many other questions in population genetics. At the same time, we can also study the genetic differences between patients and the normal ones to find the genetic mutations responsible for some of the diseases. In addition to the human genome, over 1,000 trillion base pairs from over 165,000 species have been sequenced by the end of 2013. Just reading them would take 30 million years. Life science data is not only big in size but also growing exponentially. This figure shows the number of nucleotides sequenced and stored in the GenBank database over a year. The number of nucleotides have been increasingly exponentially since 1982, doubling every 20 months. What does this mean? It means that the data deposited into GenBank in the next 20 months from now on is as much as all the data that have existed in the history of mankind till today. At the same time, the sequencing costs have been decreasing every year. The past few years have seen even steeper growth of sequencing data. The reason is that next generation sequencing technologies have been developed and used more and more widely in all areas of life sciences. Next generation sequencing technologies can sequence one person's genome in a day with less than $2,000. Just think about what exciting discoveries these never-seen-before data may contain. Other than sequencing data, other high-throughput technologies such as proteomic, metabolomic, and etc. had also generated a large variety of data. With great opportunities come great challenges. The huge amount of data requires efficient methods. Exponential growth requires scalable methods. The low signal-to-noise ratio requires accurate methods. And handling multiple types of data requires data integrative methods. These are significant technical challenges, but it depends on how you look at it. Technical challenges can also mean opportunities for technical innovations. The birth and growth of the field of bioinformatics has been driven by these opportunities and challenges. As you may be able to realize by now, the integration between life science and computer science is inevitable, and the result is the birth and growth of the field of bioinformatics. Bioinformatics can be defined as interdisciplinary field that develops and applies computer and computational technologies to study biomedical questions. It has two roles. As a technology, bioinformatics is a powerful technology to manage, search, and analyze big data in life sciences. These data can be patient statistics, telling us what is making people sick and what medicines are working, or information that allows us to create images of things too small for even the most powerful microscope to see, allowing us to study how they behave. Computers can search through all this information quickly, finding what causes diseases, helping us find new treatments, and understand how complex living organisms works. As a methodology, bioinformatics is a top-down, holistic, data-driven, genome-wide, and systems approach that generates new hypotheses, finds new patterns, and discovers new functional elements. It complements traditional experimental biology methods. A seamless combination of computational and experimental methods should be the best way to study a biological question. Bioinformatics is truly interdisciplinary. It studies questions in biology and medicine, while developing and applying methods in computer science, mathematics, statistics, and physics. It overlaps the medical and clinical informatics, systems biology, and synthetic biology. The bio in bioinformatics signifies the biological question it studies, many of which can be grouped under the conceptual framework from genotype to phenotype. Some example may help you understand better. For instance, sequence alignment. Are the sequences of two genes or proteins similar? Could they be homologous? How can we find the closest homologue of the gene we are studying from the vast databases? Can I use the known function of a known gene to guide the study of my gene of interest? Given DNA and genome sequences, how can we find the genes in the vast genome? How can we compare the similarity and difference between two whole genomes and reconstruct the evolutionary history? What are the synthetic regions between two genomes? How can we identify which genomic regions are methylated? At the level of RNA expression, we often need to find out which genes are differentially expressed between two organs or tissues, between tumor and normal tissues, or between two different developmental stages. At the level of proteins, how do you identify the expressed proteins from mass spec data? Proteins do not exist in a linear form in nature. Instead, they are folded into beautiful three-dimensional structures in nature. How can we predict the three-dimensional structure of a protein from its sequence? Molecules do not exist or function in isolation. Instead, they form complex molecular networks. How can we construct protein-to-protein interaction networks, transcription regulation networks, and metabolic and signal pathways? What are the dynamic features of these networks? Furthermore, with this data and research, how can we simulate a virtual cell. Last but not the least, how do you compare different people's genomes and use population genetic approaches to compare the genomes of the ill and the healthy and identify the gene mutations that cause diseases? From these examples, we can see that there are many interesting and important questions in life sciences that await our bioinformatics investigation. Seeing bioinformatics from another angle, the informatics in bioinformatics signifies the information and computational methods to manage, search, and analyze the data. The theme runs along the axis from data to discovery. First, the storage, index, and search of terabytes to petabytes of big data requires advanced databases. Just imagine, your laptop probably has several gigabytes of hard disk. However, just one set of our experiments often generates one or more terabytes of data, which is too large to fit in your laptop. Managing such big data requires advanced database systems. Analysis of noisy big data requires the development of many algorithms, software tools, and web servers. These make up a big portion of bioinformatics research. A tradition in bioinformatics since the early days till today have been to make most algorithms and software open source. This has contributed significantly to the broader life science research and has managed to advance bioinformatics itself as well. With these tools, we can do a lot of data mining to discover new patterns and phenomena in life science. Finally, by integrating of data and tools, we can build predictive models of biological systems. As more and more high-throughput technologies are used in life science, there will continue to be numerous new opportunities for technology innovation in bioinformatics. To meet this challenge, a new breed of professionals that could collaborate and communicate with biomedical researchers, clinicians, quantitative scientists, and bioethicists is needed to translate the genomic knowledge to improve patient outcomes. There are three main important people in the field of bioinformatics. One is biologists, who will be doing lab experiments and collecting molecular data such as DNA and protein sequences, gene expressions, etc. Another is a computer scientist, including mathematicians or statisticians, which will be helping in developing tools, software, and algorithm to store and analyze data. Third one is the bioinformatician, who will be studying biological questions by analyzing molecular data. The basis of bioinformatics is the central dogma. The central dogma is a term that Francis Crick first coined in 1958 and used to describe the fundamental process in life involving the flow of information from DNA to proteins to make a functional product. The information present in a DNA is essential to make up all proteins, and RNA acts as a messenger that carries information through the ribosomes. A DNA molecule isn't just a long, boring string of nucleotides A, T, G, and C. Instead, it's divided up into functional units called genes. Each gene provides instructions for a functional product, that is, a molecule needed to perform a job in the cell. In many cases, the functional product of a gene is a protein. For example, Mendel's flower color gene provides instructions for a protein that helps make colored molecules or pigments in floral petals. For the overview of bioinformatics, basically you start from resources. Resources include databases which are storing data in different files like nucleotide or DNA sequences or protein sequences in FASTA format. The classic data of bioinformatics includes the primary data, which is either a DNA sequences of genes or full genomes or amino acid sequences of proteins. Then, a motif, which is a super secondary structure of a protein, which can be either an alpha helix or beta sheet. Also, this secondary structure is formed to neutralize the natural polarity of different amino acids in the primary protein structure, which is a sequence of amino acid. Then, a domain in protein structure is the tertiary structure of a protein. Moreover, it forms, exists, and functions independently from the other components of the protein. Although a motif is formed through the interaction of secondary structural elements in a protein, the interactions that occur between secondary structural elements are stronger in a domain. The number and arrangement of protein domains are referred to as quaternary structure. Quaternary structure is an important protein attribute that is closely related to its function. Proteins with quaternary structure are called oligomeric proteins. Oligomeric proteins are involved in various biological processes such as metabolism, signal transduction, and chromosome replication. The goal of bioinformatics is to integrate sequence, 3D structure, expression patterns, interaction and function of biomolecules to gain a deeper understanding of biological mechanisms, processes, and systems. Bioinformatics aims to bridge the gap between data and knowledge. According to the National Institute of Health of the United States, bioinformatics deals with the development and application of analytical data and theoretical methods, mathematical modeling, and computer simulation techniques to study biological, behavioral, and social systems. The programs use public or private databases with restricted access or with economic value that have been created with information that is constantly growing and managed by institutions from various sectors. The main databases used are the following. Primary databases contain original biological data. They are the archives of raw sequence files or structural data submitted by the researchers, for example, GenBank and Protein Data Bank. Secondary databases contain information processed computationally or manually curated information retrieved from the primary databases. Translated protein sequence databases contain the functional annotation belonging to this category. For example, SwissProt and Protein Information Resources. Specialized databases are those that serve a particular research interest. For example, Flybase, the HIV sequence database and ribosomal database project are examples of databases that specialize in a particular organism or a certain type of data. Many of the problems detected in scientific research lie in the need to connect secondary and specialized databases to primary databases. It is desirable that entries in the database be cross-referenced or linked to related entries in other databases that contain additional information. Next is retrieval of data, which includes the retrieval tools or submission tools from which we are retrieving data, or we can submit the data through databases. The amount of biological relevant data is increasing so rapidly. Knowing how to access and search this information is essential. There are 3 data retrieval systems of particular relevance to molecular biologists, such as Sequence Retrieval Systems, or SRS, Intrez, dbGet. These systems allow text searching of multiple molecular biology databases and provide links to relevant information for entries that match the search criteria. The 3 systems differ in the databases they search and the links they have to other information. BankIt is a web-based form for submission of a small number of sequences with minimal annotation to GenBank. Sequen is more appropriate for complicated submissions containing significant amount of annotation or many sequences. It's a standalone application available on NCBI's FTP site. Sakura is a submission tool in DNA Data Bank of Japan, or DDBJ. Next is the query. Now, based on the sequences we retrieve or we can submit sequences, we can generate a query sequence on which we can apply multiple processes like sequence alignment. What is sequence alignment? Sequence alignment is a way of arranging the sequence of DNA or protein to identify regions of similarity that may be a consequence of functional, structural, or evolutionary relationships between sequences. Pairwise sequence alignment methods are used to find the best matching piecewise, either local or global alignments of two query sequences. A local alignment aligns a substring of the query sequence to a substring of the target sequence. It finds local regions with the highest level of similarity between the two sequences. In global alignment, an attempt is made to align the entire sequences. Global alignments are usually done for comparing homologous genes, like comparing two genes with the same function, for instance, in human versus mouse, or comparing two proteins with similar function. By contrast, multiple sequence alignment, or MSA, is the alignment of three or more biological sequences of similar length. From the output of MSA applications, homology can be inferred and the evolutionary relationship between the sequences can be studied. If we want to know how similar a mouse is to human, we need sequence alignment. According to the National Human Genome Research Institute, the genetic similarity between a human and a mouse is 85%. The figure shows a multiple sequence alignment of 5 mammalian histone H1 proteins produced by clustal O. Sequences are the amino acids for residues 120 to 180 of the proteins. In evolutionary biology and genetics, conserved sequences refer to identical or similar sequences of DNA or RNA or amino acids or proteins that occur in different or same species over generations. These sequences show very minimal changes in their compositions or sometimes no changes at all over generations. Residues that are conserved across all sequences are highlighted in gray. Below the protein sequences is a key denoting conserved sequence in asterisk, conservative mutations in column, semi-conservative mutations in period, and non-conservative mutations in space. A conservative replacement, also called a conservative mutation or a conservative substitution, is an amino acid replacement in a protein that changes a given amino acid to a different amino acid with similar biochemical properties. Completed in 2003, the Human Genome Project, or HGP, was a 13-year project coordinated by the U.S. Department of Energy and the National Institutes of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, and others. Project goals were to identify all the approximately 20,500 genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, store this information in databases, improve tools for data analysis, transfer related technologies to the private sector, and address the ethical, legal, and social issues that may arise from the project. Though the HGP is finished, analysis of the data will continue for many years. From the sequence that we align, we can construct a phylogenetic tree. What is a phylogenetic tree? A phylogenetic tree, also known as phylogeny, is a diagram that depicts the lines of evolutionary descent of different species, organisms, or genes from a common ancestor. Phylogenies are useful for organizing knowledge of biological diversity, for constructing classifications, and for providing insights into events that occurred during evolution. Furthermore, because these trees show descent from a common ancestor, and because much of the strongest evidence for evolution comes in the form of common ancestry, one must understand phylogenies in order to fully appreciate the overwhelming evidence supporting the theory of evolution. Tree diagrams have been used in evolutionary biology since the time of Charles Darwin. You can check the Open Tree of Life through this link, where you can find around 2.4 million species. Protein structure prediction is the inference of the three-dimensional structure of a protein from its amino acid sequence, that is, the prediction of its secondary and tertiary structure from primary structure. Having a protein structure provides a greater level of understanding of how a protein works, which can allow us to create hypotheses about how to affect it, control it, or modify it. For example, knowing a protein structure could allow you to design site-directed mutations with the intent of changing function. Correctly predicting protein folds based on the amino acid sequence could revolutionize drug design and explain the causes of new and old diseases. Bioinformatics has not only become essential for basic genomic and molecular biology research, but is having a major impact in many areas of biotechnology and biomedical sciences. The main uses of bioinformatics include: bioinformatics plays a vital role in the areas of structural genomics, functional genomics, and nutritional genomics. It covers emerging scientific research and the exploration of proteomes from all over level of intracellular protein composition or protein profiles, protein structure, protein-to-protein interaction, and unique activity patterns, for example, post-translational modification. Bioinformatics is used for transcriptomic analysis, where mRNA expression levels can be determined. Bioinformatics is used to identify and structurally modify a natural product, to design a compound with the desired properties, and to assess its therapeutic effects theoretically. Bioinformatics is playing an increasingly important role in almost all aspects of drug discovery and drug development. And bioinformatic tools are very effective in prediction, analysis, and interpretation of clinical and preclinical findings. Bioinformatics is applied in drug development. Drugs target only 500 proteins. Now, using computational tools of bioinformatics and disease mechanism, scientists identify new and validate drugs. Bioinformatics can also be applied in crop improvement, where collection and storage of plant genetic resources and wise application of bioinformatics help to produce stronger and more drought disease and insect-resistant crops, and improve the quality of livestock, making them healthier, more disease-resistant, and more productive. Complete microbial genome has been sequenced. It has many applications like health, energy, and industrial applications. By using bioinformatics, those genes have been isolated, which enables them to survive in extreme environments. In the not-too-distant future, with the use of bioinformatic tool, the potential for using genes themselves to treat disease may become a reality. Gene therapy is an approach used to treat, cure, or even prevent disease by changing the expression of a person's genes. Bioinformatics has major impact on biotechnology and its application. It is used to understand genes and also the genome. Bioinformatics programs are also used to compare the gene pair alignment, which help in identifying the functions of the gene and also genome functionality. Understanding evolution helps us to solve many biological problems that impact our lives and also improve the quality of life. Bioinformatics tools show a path to study evolution with a comparison of comparable genome data. Using bioinformatic tools, scientists may compare proteins or nucleic acid by using these techniques. Bioinformatics can also be applied in veterinary sciences. Bioinformatics has advanced courses of research and future veterinary vaccines development because it has provided new tools for identification of vaccines target from sequenced biological data of organism. Bioinformatics can also be applied in waste cleanup. Deinococcus radiodurans is known as the world's toughest bacteria, and it is the most radiation-resistant organism known. Scientists are interested in this organism because of its potential usefulness in cleaning up waste sites that contain radiation and toxic chemicals. Bioinformatics has provided fundamental ways of modeling a biological cell system and docking proteins that enable scientists to discover effective drug strategies to combat the diversifying problem of antibiotic resistance among casualties of infectious diseases. Many plants have been made insect resistant by incorporating the desired genes. Bacillus thuringiensis is a bacterial species that increases soil fertility and protects the plants against pests. Researchers map its genome and incorporate the potential useful genes into the plants to make them insect resistant. Corn, cotton, and potatoes have been made insect resistant so far. Bioinformatics can be applied in climate change studies. To solve this issue, bioinformatics may help by way of sequencing of the microbial genome, which reduce the level of carbon dioxide and other greenhouse gases. This plays an important role in stabilizing global climate change. In summary, bioinformatics has many practical applications in different areas of biology, medicine, and other fields of sciences that makes our life easier. And as technology improves, so do the possibilities.